So we're still covering passive transport. We finished with diffusion, molecules that just easily pass through the phospholipid bilayer are nonpolar molecules that are lipid soluble, like oxygen and carbon dioxide. And they're going to just, we just call that diffusion. But when molecules move toward their lower concentration, but those molecules require a carrier protein, then we call that facilitated transport. It's still a passive process. No energy is required. And examples are glucose and amino acids. These are molecules that are polar. They're water soluble. So they must combine with a carrier protein to be moved across the membrane, but they're still going to follow their concentration gradient. They're going to move from where their concentration is high to where it's low. And this is what facilitated transport kind of looks like. If you look at the outside of the cell, at these green solute molecules, their concentration is high outside the cell and low inside the cell. But because they're polar, they're having to move into the cell toward their lower concentration with a carrier protein. No energy is required for this, but it is very similar to diffusion otherwise. It's a passive process, and it's called facilitated transport. Now we move on to section 5.3, which is actually where I'm going to stop this lecture. And 5.3 is deals with active transport and then bulk transport. So active transport is the movement of molecules against their concentration gradient. So they're being moved toward their high concentration rather than the other way around. You can still, their carrier proteins are still used, but ATP is required. ATP is required in order to give the carrier protein the energy to pump those molecules toward their high concentration. So probably the most well-known example, the best example in biology of active transport is the sodium-potassium pump. We see this in human anatomy and physiology when we learn about muscle contractions and nerve impulses, because all of these involve the sodium-potassium pump is required to maintain a certain concentration, a certain voltage across the plasma membrane. And a certain concentration of sodium and potassium ions inside and outside the cell. So ATP is used to move 3 sodium ions out of the cell and 2 potassium ions into the cell. Both of these are being moved against their concentration gradient. So this is a picture of the sodium-potassium pump. You can see that it has 3 kind of shapes or spaces for sodium ions and then 2 spaces for potassium ions. You can see the concentration of sodium. Actually, I think the picture is misleading here about the concentrations, but you're just going to have to take my word that the concentration of sodium is high outside and the concentration of potassium is high inside the cell. So we'll look at this entire process, and what we're going to see is that sodium is going to be moved out of the cell. Look at this picture here. 3 sodium ions are moved out of the cell using ATP. ATP had to combine with the sodium-potassium pump to give it the energy to move the 3 sodium ions out. And then 2 potassium ions are moved in. And this process continues even though the concentration of sodium is already high outside and potassium is already high inside. So sodium is being moved to where its concentration is already high, and potassium is being moved to where its concentration is already high. The picture is misleading because it shows sodium concentration high inside the cell and potassium high outside. And I think the only reason they do this is I honestly think that the person who illustrated this just messed up. But you have to remember that the sodium is being pumped out, even though sodium is high outside. It's being pumped toward its high concentration. And potassium is being pumped in, even though its high concentration is inside the cell. Potassium levels are already high inside the cell, but it still keeps being pumped into the cell. So that's against both of their concentration gradients and using energy. Now we're going to learn about bulk transport, which requires the formation of a vesicle. The vesicle is composed of a phospholipid bilayer, just like the plasma membrane, because either it's going to pinch off from the plasma membrane to form, or it's going to fuse with the plasma membrane. So one thing that's interesting about endocytosis and exocytosis is that they either involve removing part of the plasma membrane to form the vesicle, or adding to the plasma membrane when the vesicle fuses with it. So in exocytosis, a bulk amount of material leaves the cell when the vesicle fuses with the plasma membrane and secretes its contents outside the cell. And endocytosis is opposite. That is when a cell engulfs a bulk amount of material or substance into a pouch, and that pouch pinches off and becomes a vesicle, and that causes the plasma membrane to actually become smaller because it removes a small portion of the plasma membrane. Depending on the type of endocytosis, there are 3 different names for it. So when material moves into the cell, it's endocytosis, when it comes in through a vesicle. But if it's large and solid, then we call it phagocytosis. For example, when human white blood cells engulf viruses or debris or bacteria, we call that phagocytosis. In fact, those human white blood cells are called phagocytes. Big ones, large ones, are called macrophages. Pinocytosis is when the cell is taking in small particles that are dissolved in a liquid. So we actually call it cell drinking, whereas phagocytosis is sometimes called cell eating. Now, if the endocytosis involves receptors, then we call it receptor-mediated endocytosis. It's a specific form of pinocytosis that uses receptor proteins and a coated pit, and we'll look at a picture in just a minute. In the genetic disease hypercholesterolemia, or high cholesterol, genetic high cholesterol, the receptor-mediated endocytosis that typically brings in the cholesterol molecules is defective. So they stay in the blood and the blood cholesterol levels remain high. So this is an image of exocytosis, and if you do your Chapter 5 homework, which I hope all of you do it, there's a good video on endo and exocytosis and then some questions that go with it. So there's a good video tutorial because it really, the process is harder to show in a picture than in a video where you can see it kind of flow. But anyway, just imagine that you've got a vesicle inside the cell and it contains this solution, these green solute particles dissolved in a watery solvent. That vesicle will move to the plasma membrane and fuse with it, and then eventually the plasma membrane will open up and secrete the substance outside the cell. We call that exocytosis, and as you can see, the plasma membrane actually gets larger because it gains more membrane when that vesicle fuses with it. And with endocytosis, you have, like in phagocytosis, you have this solid particle. Maybe it's a food particle, and maybe this cell belongs to the intestine. The small intestine is taking in a food particle, and it's going to digest it, break it down. But anyway, the plasma membrane kind of forms a pouch around that food particle, and then that pouch pinches off and becomes a vesicle that surrounds the food. And that is called phagocytosis. When that same process forms a vesicle around a liquid that has small solute molecules dissolved in it, we call it pinocytosis, or cell drinking. Phagocytosis is cell eating. And as you can see, receptor mediated means that the particles that are being brought into the cell combine with receptors on the plasma membrane, and then they form a vesicle, and that vesicle is called a coated vesicle because it's coated with these receptors that have these molecules bound to them. So that is specific for specific substances like cholesterol. Now, in this modifications of cell surfaces, it just kind of talks to you about structures that are inside the plasma membrane and outside the plasma membrane that have different functions. And we're going to skip this part. I'll mention a few of them. You've got the integrin, which is a protein that one of those plasma membrane proteins that's embedded within the phospholipid bilayer, and it can attach to these actin filaments inside the plasma membrane that help stabilize the plasma membrane and give the cell its shape. We also have these structures called proteoglycans, which have protein and carbohydrate component. We have protein fibers called elastin that give tissue its elasticity. We have collagen that gives tissue its strength. We have fibronectin, which will connect some of the fibers to the integrin molecule. So this is just a way of attaching our cells, our plasma membranes, to the outside. We have junctions between cells. There are junctions called adhesion junctions that are very strong connections that connect cells to other cells and form tissues. And then there are gap junctions that have channels between them, so that even though they connect cells to each other, they also allow for the transport of ions. Here's a picture of one of those adhesion junctions called a desmosome. These tight connections that connect skin cells together, so that if you get a sunburn and you shed some of your skin, it peels off or sheds in sheets because you have cells that are attached so strongly by these desmosomes. You have these tight junctions that form a tight seal, so that like, for example, the cells that line your urinary bladder, you don't want urine to escape into the rest of your body cavities. So tight junctions prevent that. And gap junctions allow for the passage of ions, so that ions can flow from cell to cell very quickly, and a group of cells can move in a coordinated way. They can move together, like all of the cells, all of the muscle cells in your heart, all of those cardiac muscle cells. You don't want them contracting at different times. You want them contracting in a coordinated manner so that the entire heart organ contracts at one time. And that is due to gap junctions. And cilia move together. They move in a coordinated way. They move together because of gap junctions. There is a little bit of information about plant cell walls, and the connections between plant cell walls are called plasmodesmata, and they allow for water and other materials to pass from plant cell to plant cell. Here's a picture of those plasmodesmata. And that is that finishes up our Chapter 5 lecture.